Hello and welcome to this Ask the Expert webcast. Before we get started today, we have some poll questions that our speaker would like you to answer. So on your screen, you should see a few questions. You can go ahead and click and select the answers. So the first question is, please indicate all points in your downstream purification where you use pre-packed columns. Select all that apply. And the options are early process development, process scale-up, process validation, manufacturing, or we don't currently use pre-packed columns. Okay. The second question is, removal of column packing and self-pack column hardware restrictions streamline your overall downstream development and manufacturing programs. Choose one. You strongly agree, you disagree, or you strongly disagree. And the last question is, what are the minimum benefits that motivate your adoption of pre-packed columns? Select all minimum requirements that apply. Flexibility in column size, diameter bed height, consistent packing quality, plates asymmetry, ability to connect aseptically to chromatography skid, easier tech transfer to the CMO, easier scale up, or time savings in manufacturing suite time and personnel time. So if you can go ahead and finish answering those questions, I can see that there's a few of you that haven't taken that opportunity. So please go ahead and answer the questions. We'll give you a couple more seconds, and then we will begin the presentation with Fletcher Malcolm from Repligen. Okay. So, hello and welcome to this BioProcess International Ask the Expert webcast. I'm Leah Rosen, the online editor for BioProcess International. And before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions on the question and answer window. Our questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Fletcher Malcolm from Repligen.
Thank you, Leah. Welcome, everyone. Today, we'll talk about how OPUS pre-packed columns can fast-track gene therapy development and simplify downstream operations. I am Fletcher Malcolm, and my role at Repligen in product management is product line leader for our chromatography products. And so today, today's presentation, we'll just have a brief Repligen overview, touch on some solutions to meet common gene therapy challenges, how pre-packed columns can fast-track scale up from development to manufacturing, and then wrap up with a case study. So at Repligen, we focus on bringing new technologies to market, which meet specific needs, typically unmet needs in the industry. So some of those examples are up here on the slide. And of course, my favorites are OPUS 45, 16, 80 centimeter columns, for example. These were first to market sizes for large scale purification of biologic drugs. Today, we're focused on gene therapy with this presentation. And we see some common manufacturing challenges, which are listed here on the slide in gene therapy when working with our customers. And these can be meeting the demand from upstream bioprocessing. And so with low binding capacities that we typically see in gene therapy applications on the capture step and steps, meeting the demand is sometimes a critical challenge. Reducing time to market, this can be accelerating what comes in early stage development, how fast can we then get it into the clinic? And then ensuring product quality and safety. And so for our pre-packed columns, the flexibility that we have in column size, resin type, et cetera, and the scalable platforms that we have from early process development to manufacturing, help our customers implement these technologies to be able to run their processes effectively. Reducing time to market using our innovative robo-columns or our mini-chrome columns. These columns can be used for rapid process development and then scale up to GMP manufacturing. And a little bit more unique to the gene therapy marketplace, enabling closed processes with tubing assemblies to reduce or eliminate as much bioburden risk as possible is something that we see is becoming more and more common. And so we'll talk a little bit about that later in the presentation. The journey that many of our customers go through, and this is not unique to gene therapy, is starting out at small scale, perhaps doing automated high throughput experiments with robo-columns, or maybe it's semi-automated with columns such as mini-chrome columns with a variety of different resin types, screening different conditions to figure out which combination of resin and chromatography method conditions might work the best. And then from there, scaling up, perhaps doing process characterization or validation, and then taking that to GMP production. And at Repligen, we've recently studied the scale-up effects from small to large. And this was a study that was published in the Journal of Chromatography A last year. And this particular example from that reference, that paper, looks at lysozyme loaded to breakthrough. And you see the breakthrough curves on the left. And what you notice is that there is a similarity when you're going from 5 by 5 centimeter diameter columns. So those are mini chrome. And that's a 1 mil column all the way up to a 45 by 20 centimeter column. And so what you're looking at is a good consistency between the different column sizes as you scale. No diameter is changing, even though bed height is changing in some cases. We see that good consistency, which is showing that packed bed stability is good, as well as the column hardware effects as those changes occur from large to small are not impactful. There are small variations, as you can see. These were studied and attributed to slight conductivity differences in the load solution. So the reason why those curves don't line up perfectly. And then on the right side of the slide, you see the binding capacity, consistent binding capacity from small columns all the way to the larger columns. So this it makes our jobs much easier when we're thinking about scale up from small scale to large scale. We no longer have to worry about things like packing methods or self-pack column features. We can eliminate those as variables and focus on the things that we need to do to make our process successful. And so that can also eliminate GMP suite time for packing, something that we see as a common bottleneck in many of our customers. So again, eliminating some of these things that are here on the slide to allow you to focus on what's most important in developing your process, not necessarily the nuances of column packing. And so when we talk to customers about what makes their GMP purification successful, or what will make their GMP purification successful, we commonly hear these two top items in the gene therapy world. One, closed processes for bioburden control, and two, flexible and platformable technology. That meaning that we can use the same column type, no matter what the resin size, and we have the ability to configure different bed heights, resin types, diameters, etc. So with OPUS pre-packed columns, we make that easy. A large number of diameters available, bed heights which can be configured to the nearest millimeter, and we can pack with resin from any supplier, and that includes custom resins, which are becoming more and more popular. And then finally, these are typically delivered within 4 to 6 weeks from resin arrival at Repligen. Even though the columns are built to order, our operations are optimized to have a quick turnaround to meet our customer requirements. Just a case study here. This is an ongoing application and customer that we work with. They came to us originally a few years back with this applications profile. So one single small volume affinity chromatography step. In this, just one capture step. Sometimes we see, depending on the application in gene therapy, sometimes there's a capture and then one polishing step, but this one just uses a single capture step. Binding capacity is relatively low, but they did employ a custom resin for increased specificity for their target molecule, and came to us with the request for a closed or aseptic process for microbial control. And so one of the challenges was there was a large number of columns were needed, so greater than 100 columns. They all needed to be connected aseptically. And one column would be used for each batch, which means that a column failure would essentially mean a batch failure, and that, of course, wouldn't be acceptable. And so we needed to develop a robust packing method. And the end solution needed to be easily transferred from their facility to a CMO for most of the production that would occur for clinical, early stage clinical trials. So the solution, we used OPUS, customer used OPUS mini chrome columns for process development, and then scaled up to an OPUS 5 with a 250 mil column volume. And we designed a customized tubing assembly. There's a schematic of that on the right, which would come attached to the column when it arrives at the customer's facility. And that enables them to connect to their skid aseptically, so that it's easy to use and as low bioburden as possible. And so from our side, each column was packed and performance tested, results printed on the C of A. And so just to give you a little bit of a snapshot of what we did from a column packing standpoint, this slide shows some of the process control data from column packing in our facility for several columns packed between Q4 2019 and Q1 of 2020. And what you see is some variation in the results from efficiency and asymmetry, which are on the left. But in all cases, the results are either above spec in the case of efficiency or well within spec in the case of asymmetry. Given that this is a capture step, the variations are not significant, and I would say relatively normal for many of our processes. Importantly, with this particular resin, needed axial compression packing, which we were able to do. And after that axial compression, where the flow distributor consolidates the beads at the target bed height, we conditioned the columns to a very consistent target packing pressure, as you can see, just above 4 psi. And this led to consistently packed columns that could be relied on by the customer. And then lastly, of course, after packing, there's a sanitization step. And as you can see, there's near zero bioburden detected post-sanitization for all columns. And so why is this significant? In addition to making sure that the columns work when they arrive, there's significant time savings for the customer if they can justify simply releasing the column on C of A. So because we test every single column that we pack, and every single column has a unique C of A, the columns can easily be released using a certificate of analysis with little to no worry about packed bed stability in transit. And so shipping validation is something that we have studied time and time and again. There are some results on the right. Packed bed stability is not a challenge with transit. Therefore, you can take a risk-based approach of not testing for plate count and asymmetry, which means when the columns arrive at an end user's facility or your facility, which means there's no additional buffer prep, there's no production downtime for testing, because typically the same equipment would be used for testing these columns as well as running them. There's no qualification batch records to review. There's no deviations that need to be recorded or reviewed. These are all things that would be taken care of by us. And this, as shown on the left-hand side of the screen, saves about 4 hours of suite time and 16 to 20 FTE hours per column. So with 100 columns, that's 400 suite hours of suite time and 1,600 to 2,000 FTE hours. And that's very, very significant if you think of this campaign. So with pre-packed columns becoming more and more used in the industry, we do see a movement away from some of this incoming qualification testing, especially for repeat orders such as this one. So finally, just to wrap up, as we look here at pre-packed columns, how they can help your operations, there's easy implementation and operations. Columns can arrive, be and run in a matter of hours. And we simplify your processes by eliminating all those packing concerns, column testing concerns, the deviations that might be recorded associated with those, again, to streamline the operations, to fast track your results. And then ensuring the quality and reliability is a key part of what we do. Our state facilities in Waltham, Massachusetts, in Ravensburg, Germany, for packing columns ensures that columns will arrive ready to use and makes your job easy when you're thinking about developing processes from early stage to late, or well, hopefully late stage, and being able to use one consistent column packing platform throughout the lifetime of that molecule. So thank you very much, and I will take any questions.
Okay, great. Thanks, Fletcher. So the first question is, for incoming quality control, is it a requirement for me to test column performance prior to use?
Right. So so we get this question from customers, and the answer is it depends. There is no specific requirement that says you have to test for plate count and asymmetry when the column arrives at your door. Many customers like to at least have an understanding when they first choose a column, first choose an OPUS column, for example. Maybe they'll test or do some surveillance testing in a pilot or engineering setting, and then decide whether or not that needs to be incorporated from an incoming materials assessment for manufacturing. So we can and have seen many companies take the risk-based approach that no incoming qualification testing for chromatography does not need to be done. We can simply use the results on the certificate of analysis. Okay.
And what sanitization solution was used? Is there a specific time period for sanitization hold, and can these columns be gamma irradiated?
Yeah, that's a very interesting question. So sanitization is typically specific to the resin type. So I don't remember with that specific resin what the exact sanitization solution or exact sanitization procedure was. Sometimes they can include a hold. Sometimes they're more dynamic. Again, depends on the requirements for the resin, and that sanitization procedure, if required, especially in the case of a custom resin, would most likely be customized to that end user's needs. And those are all things that we can do. And then to answer the follow-up question is, can the columns be gamma irradiated? The answer is yes, they can be gamma irradiated. And that is something that some customers have requested. We do offer that as essentially a custom service, but today is not the norm in gene therapy applications.
Okay, and can the media be repacked in the same column, and if so, how many times?
So once the resin is packed in the columns, generally it's there for the life of that resin. So unpacking the columns requires destruction of the column hardware, and while that can be done, we wouldn't necessarily recommend it. And then because it would be essentially destructive. You'd have to cut the column open with some type of saw. It was not something we would recommend. So once the columns are packed, the resin is generally in that column for good. So the question would be, why would unpacking be required? Is it because we're worried about maximizing resin lifetime, or is it because we want to remove the resin after the columns may have been used for GMP and use that resin for some sort of non-GMP activity? So yeah, we understand the reasons why somebody may want to do that, but there is no easy way to unpack that resin.
Okay. And what is your recommendation to test bioburden for protein A long-term storage with 20% ethanol once a year or every 6 months?
So testing for bioburden after long-term storage, I think I'm not an expert in this area, but what we see our customers do is typically after long-term storage, and again, it depends, is that long-term storage after the column has been used or unused? So if the column has arrived from Repligen and it's unused, I would say that you could always run a sanitization, but we've shown and have data that there's no bioburden growth when the columns sit on the shelf. So it could be done. I don't think it needs to be done. It's a little bit different story if the columns have been used in GMP production and then put into storage and then pulled back out. In that case, we do see end users often testing for plate count asymmetry after storage and also doing sanitization cycles and then subsequently testing for bioburden. So again, I think it's sort of an it depends, but what we have shown is that when the columns are sanitized properly and when they're stored in a bacteriostatic solution, the bioburden doesn't grow. So it would be important just to consider your own process and what you think you need to do to make sure that the bioburden control is sufficient.
Okay, great. Thanks, Fletcher. And if we didn't get to your questions, we are passing all the questions along to Fletcher, and he can follow up with you directly. So thank you to our audience for joining us. The recorded version of this webcast will be made available for replay in the multimedia section of our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.